Axonal transport can be studied by visualizing the larval segmental nerves of third Instar Drosophila. The larva is first dissected, exposing the CNS without damaging the optical lobes, ventral ganglion, and segmental nerves. The second step of the procedure is to fix the larva in formaldehyde before incubating with primary antibody overnight and the secondary antibody for an hour in the dark. The larva is then mounted on a glass slide, and axonal transport defects, protein localization, and protein distribution can be examined through immunofluorescence microscopy.
Though this method can provide insight into axonal transport, it can also be applied to other systems such as synapse and muscle morphology.
Generally, individuals new to this method will struggle because initially it's difficult to perform this larval dissection, especially to keep the brain, segmental nerves, and the muscles intact.
To begin this procedure, prepare 1 times dissection buffer, fixative, 1 times PBT, and 5% BSA solution as detailed in the table shown. Before the dissection, gather a clean pair of number 5 forceps, a clean pair of straight blade Vannis scissors, and pins. To start the dissection, use a microspoon to collect wandering third instar larvae with a specific genotype onto a petri dish. Then wash the larvae by rinsing them in a petri dish filled with deionized water to get rid of all the food.